Now we'll come to the next important part, that is the vectors that are used in your rDNA technology. Okay, vector is actually a DNA molecule which has the ability to replicate autonomously in an appropriate host cell, and in which it can easily take up foreign DNA and used for cloning purpose. So that is the definition of your vector. Okay, the most commonly used vector is your plasmid. Plasmid is nothing but an extra chromosomal DNA. It is a double-stranded circular DNA. So your plasmid looks something like this. Double-stranded circular DNA. It is present extra chromosomally and it has the ability to replicate on its own. So we will see some features of the vectors. Okay. Now there are three important things that the vector should contain. First thing is your origin of replication. Without origin of replication, the plasmid or the vector will not have the ability to grow on its own or replicate on its own. So, origin of replication is a very important feature which a vector should have. So, that is origin of replication. Second important feature that a good vector should possess is your multiple cloning site. Now this multiple cloning site contains a sequence of nucleotides in which n number of restriction endonucleases can recognize this sequence of nucleotide. Okay. Now why it is called multiple? Because you cannot be restricted for one restriction endonucleases to act on one plasmid only. Okay, so that is why it should have multiple cloning sites. Okay, third important feature is it should have one selective marker. Okay, what is the importance of selective marker is that once you have put your recombinant DNA into your host, you should be able to select the cells which contains the recombinant DNA, and that is done with the help of using a selectable marker. Okay. Now what examples of selectable marker is your maybe your antibiotic resistant site, or you can use certain new techniques like using some fluorescent tags which are attached to your plasmid. Okay. Now what is the concept of this selectable marker? Usually you can have a selectable marker which is away from this multiple cloning site, or the one which is preferred is you have your selectable marker into the multiple cloning site. So what happens is, if you have a sequence called GATTC, okay, suppose a restriction endonucleases comes here and try to break this particular sequence. Okay, so when now your plasmid is open, because it has opened, it has also broken the sequence which was responsible for antibiotic resistance. Okay, so before the action of restriction endonucleases, your plasmid was antibiotic resistant, but now because it has broken that particular sequence, your plasmid is now not resistant to your antibiotic. So when the restriction endonucleases cuts the sequence for your antimicrobial resistance, the plasmid is no longer resistant to the antibiotic. So with the help of replica plating technique, we can identify which plasmid has taken up the DNA and which plasmid has not taken up the DNA. Okay. So to summarize, your plasmid looks something like this. So it has an origin of replication. This entire thing is your multiple cloning sites. So as you can see, you can use these all restriction endonucleases all in one plasmid itself, and then you have these two sites which are called as your selective markers, which are coding for ampicillin resistance as well as your tetracycline resistance. So these are some of the features that an ideal vector should possess. Okay. Now we are going to study different types of vectors. So broadly, your vectors can be classified into two types. One is your cloning vector and second one is your expression vector. So whatever ideal properties of a vector that we have studied, those were applicable for your cloning vectors. Okay, so that means it should have an origin of replication, it should have multiple cloning site, and it should have a selectable marker. Okay, now in addition to these properties, an expression vector would need something additional. What are those additional things are? It should contain a promoter, it should contain start or stop codon for translation, transcription and translation. And it should contain some tags. Common example is your his tag. Okay. Now the basic purpose of an expression vector is to make your final product is going to be some kind of protein. Okay. So the ideal properties that we have seen in a vector, those are not enough because they don't have the ability to produce protein. Okay. So in addition to that, you need promoter which will start the initiation of protein synthesis. It should have transcription and translation, and later on when your protein is synthesized in that host cell, you should have some mechanism by which you will fish out that particular protein, and that is done by using some tagging techniques like using your histidine. So we will not talk about expression vectors in details here. We will be mostly talking about your cloning vectors. So here are few examples of cloning vectors. First one is your plasmid. You have your cosmid. Then you have your lambda bacteriophage. You have bacterial artificial chromosome and yeast artificial chromosome. So these are the different types of cloning vectors that you can use. Plasmid is the most common that is used, and that we have studied earlier.